Hi everyone, this is Reema here, your biology educator from V-Learning. So as you know that V-Learning is an online educational platform where we strive to provide quality education for all. So to get all the benefits from our channel, do subscribe to it, and also don't forget to visit to our website, that is www.v-learning.in, where you will be getting all the pre-recorded videos, the live classes and the PDF notes of all this topic. So let's begin today's session. So in our last class, we have learned about plasma membrane and the cell wall, right? So there you have seen that the plasma membrane is made up of mainly three components, that is the protein, lipid and the carbohydrates, right? And also you have learned about the structure of the cell, the structure of the cell wall, right? So in today's class, we are going to continue with the chapter number 8, the next topic, that is the endoplasmic reticulum, Golgi bodies, lysosome and the vacuole. And this is your lecture number 4. So let's begin today's session. So see, endoplasmic reticulum. So inside the cell, as you know that there are various cell organelles, right? So among them, endoplasmic reticulum is also one of the cell organelle, right? So as you can see in the picture, so this is the picture of the endoplasmic reticulum. So where you will find this endoplasmic reticulum? This endoplasmic reticulum, you will see it near the nucleus, okay? So if this is the nucleus, okay, if this is the nucleus, the endoplasmic reticulum will be present near the nucleus of the cell, okay? So it will be surrounding near the nucleus. So this is the nucleus, and you will see the endoplasmic reticulum scattered in the cytoplasm, so near the nucleus. So this endoplasmic reticulum, it is basically responsible for the synthesis of the protein and the lipid in the cell. So what is the function of this endoplasmic reticulum? It is basically responsible for the synthesis of the protein and lipid in the cell. So as you can see in the picture, here you can see this is the nucleus, right? So this is the nucleus. So surrounding the nucleus, you can find the endoplasmic reticulum. So the nucleus has got nuclear pores. So near the nucleus also will be having nuclear membrane. So here you will find the nuclear membrane. All right, this will be the nuclear membrane, and near the nuclear membrane you will see the nuclear pores too. So see, these are the ribosomes which are attached to the endoplasmic reticulum. So there are some ribosomes which are attached to the endoplasmic reticulum, and there are some ribosomes which are free in the cytoplasm. So let's see the diagram of this endoplasmic reticulum, okay? So inside the cell, as I said, that if this is a cell, all right, so this is the cell, so you will find the nucleus here, okay? So this is the nucleus, right? The endoplasmic reticulum will be scattered, okay, will be surrounding the nucleus of the cell. So this is the ER, and the Golgi bodies, okay, this is the Golgi bodies. It is also present near the endoplasmic reticulum. So in the cell, you will find this endoplasmic reticulum. This is a nucleus. The nucleus will be having the nuclear membrane too, right? This is the nucleus of the cell. So this is what? This is the nucleus of the cell. So this is the endoplasmic reticulum. Yes. All right. So just a minute. Yes. So see, this is the endoplasmic reticulum. It is present near the nucleus. So here again, different cell organelles are present, like your mitochondria, then the lysosomes, okay? Lysosomes, the ribosomes, okay? Different cell organelles are also present inside the cell. So the endoplasmic reticulum is a network which is surrounding the nucleus, and and what are the different types of endoplasmic reticulum? You will see the different types of endoplasmic reticulum. Okay, the different types of endoplasmic reticulum you will see is the cisternae, okay, cisternae, then vesicle and the tubule. So these are the three types of the endoplasmic reticulum you will find. So how is the cisternae? Cisternae is a flattened sac-like structure. Okay, so these are basically the flattened sac-like structure where the ribosomes are attached. So in the cisternae, the ribosomes are attached. So these ribosomes are responsible for the synthesis of the protein. Okay, so in the cisternae you will find the ribosome. Okay, so this cisternae are flattened sac-like structure. Now what about this vesicle? So this is the cisternae. Okay, this is the cisternae. Now what about this vesicle? These vesicles are round spherical structure. Okay, so these vesicles are round spherical structure. So here you will not see the ribosomes. Okay, so here you will not see ribosome. And what about the tubules? So the tubules looks like this. Okay, so this is how the tubules looks like. Okay, this is the structure of tubule. So these are the three types of endoplasmic reticulum, right? So in the endoplasmic reticulum, as you have seen that there will be ribosomes will be attached. Okay, so where the ribosome is attached, that is known as what? That is known as rough endoplasmic reticulum. Okay, that means what? In the endoplasmic reticulum, the ribosomes are attached. Okay, these are what? These are the ribosomes. So here the ribosomes are attached, and that is known as what? That is known as rough endoplasmic reticulum. And in the endoplasmic reticulum, for example, here you can see that there is no ribosome, right? So this is known as what? This is known as smooth endoplasmic reticulum. So this is the structure of the endoplasmic reticulum. So by now you know that there are two types of endoplasmic reticulum, right? So what are the two basic types of endoplasmic reticulum? The two types of endoplasmic reticulum are SER and RER. Okay, so what are the two types of endoplasmic reticulum? SCR and RER. So what is this SCR? SCR is smooth endoplasmic reticulum. Okay, and RER is rough endoplasmic reticulum. So we will study it in the next topic. So see, what is this endoplasmic reticulum? It is a network. Okay, it is a network or reticulum of tiny tubular structure scattered in the cytoplasm. Okay, so it is a network or a reticulum of tiny tubular structure. Okay, these are the tubular structure which is scattered in the cytoplasm. So if this is the nucleus, okay, so these are what? These are the endoplasmic reticulum which are scattered in the cytoplasm. Okay, here you will find some smooth endoplasmic reticulum. Okay, so these are the smooth endoplasmic reticulum. So some endoplasmic reticulum will be having ribosomes attached to it. So some will be having ribosomes attached to it. Okay, so as you can see, these are what? Ribosomes are attached, right? So it is known as what? It is known as rough endoplasmic reticulum. Rough endoplasmic reticulum means it is the endoplasmic reticulum where the ribosomes are attached. Where the ribosomes are attached. Now what is this smooth endoplasmic reticulum? So these are what? These are the smooth endoplasmic reticulum. Here you will not find the ribosome. So here we will not see the ribosome. So no ribosome you can write. That is, the ribosomes are not attached. Clear? So these are the two types of endoplasmic reticulum. So the ER divides the intracellular space into two distinct compartments. So what are the two distinct compartments? One is luminal, okay, and another is extraluminal compartments. Okay, so these are the two distinct compartments. Next, so as you already know, what are the two types? One is smooth endoplasmic reticulum, another one is rough endoplasmic reticulum, right? Yes. So next, let us learn about the smooth endoplasmic reticulum. So in the absence of ribosome, that means ribosome is absent, right? So they appear smooth and are called as smooth endoplasmic reticulum. So the smooth endoplasmic reticulum is the major site for the lipid synthesis. Okay. So what is the function of this smooth endoplasmic reticulum? The function of the smooth endoplasmic reticulum is lipid synthesis. Okay, lipid synthesis. synthesis of steroids, okay, synthesis of steroids, synthesis of the fat cells, okay, synthesis of the steroids, okay. So they are the major signs for the synthesis of lipid. So in animal cells, lipids like steroidal hormones are synthesized in the SCR. So in case of animals, we have various steroid hormones. So what are the various steroid hormones in case of male? In case of male, the steroid hormone is what? Testosterone, okay? Testosterone, okay? Testosterone. And in case of female, what are the various steroid hormones? Progesterone and estrogen. Okay, the spelling. Testosterone, progesterone and estrogen, these are the various steroid hormones which have been secreted with the help of what? With the help of this SCR, okay? So next let us learn the rough endoplasmic reticulum. As you know that the rough endoplasmic reticulum is what? The ribosomes will be attached, right? So the ER often shows the ribosomes attached to the outer surface. So the endoplasmic reticulum where the ribosomes are attached, okay, where the ribosomes are attached, we call it as rough endoplasmic reticulum. So the endoplasmic reticulum bearing ribosome on their surface, okay, is called the rough endoplasmic reticulum, and it is responsible for what? It is responsible for the protein synthesis, okay. So it is responsible for what? It is responsible for protein synthesis. So RER is frequently observed in the cells and involved in what? Involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. So they will be continuous towards the outer membrane of the nucleus, and they are responsible for the protein synthesis. Clear? So this is the function, or these are the function of the smooth endoplasmic reticulum and rough endoplasmic reticulum. So so you have seen that where the endoplasmic reticulum is exactly situated, what are the various two types of endoplasmic reticulum, and what are the functions of these two types, that is SCR and RER, right? So now let us move to the another cell organelle, that is the Golgi bodies, okay. So Golgi body, as you have seen, it is also present in the cytoplasm. It is also scattered in the cytoplasm near the endoplasmic reticulum, and this Golgi body is basically responsible for what? Modification of the protein. So as you have seen that the endoplasmic reticulum, that is the RER, is synthesizing the protein, right? So the Golgi body, what it does, it modifies the protein, okay. It is responsible for packaging and modification of the protein. So it was discovered by whom? It was first discovered by Camillo Golgi, okay. Camillo Golgi in the year 1898, okay. So it was first discovered by Camillo Golgi in the year 1898. So as you can see, it looks like a sac-like structure, isn't it? It is a sac-like structure which is present near the endoplasmic reticulum. So it has got various sac-like structure, okay? It has got various sac-like structure which are known as cisternae. So in each Golgi body, you will find 4 to 6 cisternae, okay? The cisternae are what? The cisternae are disc-shaped like structure, okay? The cisternae are what? These are the cisternae, as you can see, right? So these are the cisternae. They are present in the form of stelts, okay? They are present in the form of stelt, and they are disc-shaped like structure. So there are total 4 to 6 cisternae in a Golgi body. So as you can see, these are the cis phase and this is the trans phase, okay. A Golgi body has got two phases. One is the cis phase and another one is the trans phase. So in the Golgi body, what are the two phase you see? The two phase we see in the Golgi body, one is the, yes, one is the cis phase. So one is the cis phase and another is the trans phase. Okay, another is the trans phase. So the cis phase, okay, this cis phase is responsible for receiving the proteins, okay, and this trans phase is responsible for delivering the proteins to the nearby cells. So let us see how is the structure of the Golgi body. So this Golgi body, it looks like this, okay. So these are what? These are the cisternae, okay. So these are the cisternae and this has got the trans phase. This is the cis phase, okay. This is the cis phase. This is the trans phase. This is the trans phase. These are what? These are the vesicle, okay. These are what? These are the vesicle. These are the secretory vesicle, okay. So they are responsible for the process of delivering. These vesicles are spherical in shape and they are responsible for delivering the proteins to the nearby cells. And through the cis phase, the protein is being received, okay. Through the cis phase, this vesicle, it brings the protein, and here the vesicle, it modifies and it delivers the protein to the nearby cell. So this is how the structure of Golgi body looks like. So it was first discovered by Camillo Golgi in the year 1898, as you know that. So it is a densely stained reticular structure near the nucleus. So where it is present near the nucleus. So these were later named as Golgi bodies after him. So after the scientist, after the name of the scientist only, these were named as Golgi bodies. So they consist of many flat disc shaped like sacs or cisternae of 0.5 micrometer to 1 micrometer diameter. So what they consist of? They consist of cisternae, okay. And what is the size of the cisternae? It is 0.5 micrometer to 0.5 micrometer to 1 micrometer. 0.5 micrometer to 1 micrometer. So these are stacked parallel to each other and varied number of cisternae are present in the Golgi complex. So the number of the cisternae will vary from one Golgi body to another. So the Golgi cisternae are concentrically arranged near the nucleus with distinct convex, that is cis phase or the forming phase, and the concave trans or the maturing phase. So the cis phase is what, as I have said, that the cis phase is your forming phase, okay. This is the forming phase where the vesicles are being formed, okay. And this trans phase, okay. The trans phase are what? The trans phase are the maturing phase, okay. These are the maturing phase. So this cis phase, what it is known as? It is also known as what? Convex, okay. It is also known as convex, and the trans phase is also known as concave. So the cis and the trans phase of the organelle are entirely different but interconnected. So the cis and the trans phase, they are different from each other, but both are helping in the formation of the protein in the cell. That is in the process of transportation, in the process of storage and the modification of the protein. So the function of Golgi body. So what is the main function of Golgi body? The Golgi body is responsible for the storage, okay, then modification, packaging of proteins, okay. This is the main function of the Golgi body. So what is the main function? That is storage, modification and the packaging of the protein. So this is responsible for storage, modification and the packaging of the protein. So the Golgi apparatus is performing the function of packaging the materials, you can see, that is delivered to the nearby cells or to the intracellular targets outside the cell. So once the protein is been secreted, so the Golgi body is responsible for what? The Golgi body is responsible for storage, modification, and the packaging, okay, and the packaging of all the materials which are needed to be delivered to the nearby cells. So a number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from the transphase. The Golgi apparatus is the important site for the formation of glycoprotein and glycolipids. So if the protein is, that is, if the glycoprotein, what do you mean by this glycoprotein? Glycoprotein means if the protein is attached to the sugar molecule, then it is called as glycoprotein. And if it is attached to the if the lipid is attached to the sugar molecule, then it is known as the glycolipid. So it is also responsible for the formation of what? It is also the important site for the glycoprotein and the glycolipid. So where the protein is being modified, it is modified in the cisternae. So the modification of the protein, where it takes place, it is taking place in the cisternae, okay, in the cisternae of the Golgi apparatus. Yes. So this is all about the Golgi body. So in the Golgi body, you have seen how is the structure of the Golgi body, right? And who has discovered the Golgi body? Where exactly the Golgi body is present? It's present near the nucleus, right? So, and you have seen that what are the various phases of the Golgi body, like the cis phase and the trans phase, right? So cis is the form. Cis is what? Cis is the forming phase, isn't it? And trans is the maturing phase. And also we have learned about the function of Golgi body. So this is all about Golgi body. Now let us learn the lysosome. Now what is this lysosome? So lysosome is also one of the cell organelle. Okay, it is a single membrane cell organelle. So lysosome, where it is present? So if we see, this is a cell. Okay, so this is a cell. So here the nucleus are present. Near the nucleus is present. So this is what? This is the ER. Okay, this is the endoplasmic reticulum. Here the Golgi bodies, right? This is the Golgi body. So this is ER, right? Then this is what? This is Golgi body. Now this Golgi bodies are secreting the proteins, isn't it? These are secreting the proteins or the vesicles, right, with the help of these vesicles. So the smaller vesicles which are present inside the cell, that they are known as what? They are known as lysosome. Okay, these are known as lysosome. And the bigger structure which we see, this is known as what? This is known as vacuole. Okay, this is known as vacuole. So how is this lysosome exactly? How is the structure of the lysosome? Inside the cell, you will see in the cytoplasm. Okay, this area is what? This area is the cytoplasm. Okay, so these lysosomes are what? These lysosomes are the small bead-like structures which are lying in the cytoplasm. Okay, so they are basically what? They are small. Okay, they are small and they are spherical. They are small, they are spherical structure which is found in the cytoplasm. Okay, in the cytoplasm. This is the nucleus, and this area where the cell organelles are present, that is known as what? That is known as cytoplasm. So this lysosomes are small and spherical structure which is found in the cytoplasm. Now, these lysosomes are very much essential for the digestion of the cells. Okay, and they contain the digestive enzymes. Okay, so they contain the hydrolytic enzyme. Okay, they contain the hydrolytic enzyme. Okay, hydrolytic enzyme which is responsible for the digestion of protein, fats and lipids. Okay, so this lysosome has the capacity to digest what? To digest the protein, then to digest what? To digest the lipid, and to digest what? To digest the carbohydrate. Okay, carbohydrate. So how they digest the how they digest the protein? They digest the protein with the help of the enzyme proteinase. Okay, proteinase, then lipid with the help of the enzyme lipase, and carbohydrate with the help of the enzyme carbohydrates. Okay, carbohydrates. So they digest the enzyme with the help of this hydrolytic enzymes. Okay, these are the hydrolytic enzymes. So lysosome, what it contains? It contains the hydrolytic enzymes, and the pH. And the pH of the lysosome is acidic. Okay, the pH of the lysosome is acidic. So these lysosomes are also known as the digestive bags and also known as the suicidal bags. Now we will learn why they are known as the digestive bags and the suicidal bags. So yes, as you can see, they are the membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. So in this diagram, you have seen that the membrane-bound vesicular structure. So these are the vesicles, isn't it? From the Golgi body, these are the vesicles. So from the vesicles, these are formed. That is, lysosomes are being formed from the Golgi bodies. That is, from the vesicle of the Golgi bodies, formed by the process of packaging in the Golgi bodies. So the isolated lysosomal vesicle have been found to be very rich in all types of hydrolytic enzymes. So as I have said that they are rich in various types of hydrolytic enzymes. Okay, hydrolytic enzyme. So what are the various hydrolytic enzymes? Like lipase, proteinase, and carbohydrates, which is responsible for the digestion of protein, lipids, and the carbohydrates in our body. So they have got what? They have got a acidic pH. So what is the pH? The pH is acidic pH. So these enzymes are capable of digesting the carbohydrates, proteins, lipids, and nucleic acid. So this is very, very easy. This is very, very simple and easy. This cell organelle, that is lysosome. Now, this lysosome, why they are known as the digestive bags? They are known as the digestive bags because they are responsible for the digestion of the cells. So any of the cell in our body, if they are damaged, okay, if they are torn, the lysosome, what it will do? It will eat up the cell. It will digest the cell. Okay, that's why it is known as the digestive bag. Okay, digestive cell, right? Or the suicidal bags. Okay, so this is the reason that is lysosome has the capacity to eat up the damaged cell in our body. So next, let us learn the next topic is your vacuole. Okay, so vacuole is the membrane bound space found in the cytoplasm. So how is the vacuole? If you see the cell, okay, you will see a large vacuole in case of the plant cell, right? So this is what this is, the vacuole. Okay, so in case of the plant cell, the vacuole is large or small. In case of the plant cell, they have got what? They have got large vacuole. In case of the animal cell, so they have small vacuole. Okay, so they have got small vacuole. So animals have got small vacuole. Now, where it lies? It lies in the cytoplasm, as you know. Okay, so all the cell organelles, it lies in the cytoplasm. It contains what? It contains water, sap, excretory product, and other materials which are not useful to the cell. Okay, so it contains what? It contains the sap, excretory product, and other materials which are not useful to the cell. So the vacuole is bounded by a single membrane called tonoplast. So this vacuole, it is bounded by a single membrane. It has got what? It has got a single membrane which is called as the tonoplast. So what this tonoplast provide? This tonoplast provides rigidity. Okay, so this tonoplast provides rigidity to the vacuole. So in plant cells, the vacuoles occupy up to 90% of the volume. So you will see that the vacuoles of the plant cell are generally larger because they occupy the 90%. So there are various types of vacuole. So if we see the plant cell, so which looks like this, right? So the plant cell, in case of plant cell, they will be having a very large vacuole, right? In the plant cell, the vacuole will be large, right? They will be having large vacuole. Whereas in case of the animal cell, the vacuole will be small. So they have got small vacuole. Okay, the animal cell has got small vacuole. Now, there are various types of vacuoles again. Okay, there are various types of vacuoles. So, what are the various types of vacuole? The various types of vacuole are sep vacuole, then contractile vacuole, then food vacuole and gas vacuole. Okay?
Down, then up.
Vacuole. So these are the 4 types of vacuole you will find. So what are the various 4 types? Sep vacuole, contractile vacuole, food vacuole and the gas vacuole. Now this sep vacuole you will find only in case of plants. Okay? Only in case of plants you will find the sep vacuole. Then contractile vacuole you will find in case of the fish or in case of the animals. So this contractile vacuole, it is responsible for the process of osmoregulation. It is responsible for the process of osmoregulation. Now this food vacuole you will find in case of amoeba. In case of amoeba, you will find the food vacuole. And the gas vacuole you will find in case of the bacteria. In case of bacteria, you will find the gas vacuole. So these are the various vacuoles. Now, how the amoeba, you know that food vacuole, okay? So let's say in case of plant, okay? So this is the sep vacuole, okay? So this is the sep vacuole. So where you will find this vacuole? You will find this vacuole in case of the plant, okay? So sep vacuole, it consists of water, okay? It consists of resin, okay? It consists of various materials which are not useful to the plant, okay? Now, in case of this amoeba, that is the food vacuole, where you will find in case of amoeba. So you have seen how is the structure of amoeba, right? So this is like this. This is the structure of amoeba. So if there is any food, this is the food, for example. So amoeba has the capacity to capture the food, isn't it? So these are what? These are pseudopodia, right? So these are the pseudopodia. With the help of pseudopodia, they have the capacity to capture the food. So the amoeba, it will capture the food material inside it with the help of this food vacuole. So they have got what? They have got food vacuole. So there are various types of vacuole, and this vacuole also plays an important role in the body of an organism. So basically these vacuoles are responsible for the process of storage of food. So these vacuoles are responsible for the process of storage, for the process of providing turgidity, for the process of providing support to the cell. Okay, so these are the functions of the vacuole. So in plants, the tonoplast facilitates the transport of number of ions and other materials against concentration gradient into the vacuole. Hence their concentration is higher in the vacuole than in the cytoplasm. So these vacuoles are also responsible for what? Also responsible for the transportation of the ions, and the minerals. Okay, transportation of ions and the minerals in the cells in case of the plants. So where? In case of the plants. So these are what? These are the functions. Okay, these are the functions of the vacuole. So in amoeba, the contractile vacuole, okay, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in proteins, food vacuoles are formed by engulfing the food particles. So as you have seen that in amoeba, it is responsible for capturing the food, right? So they have got various functions. That is, the vacuoles also plays an important function in the inside the cell. So this is all about today's session. So I hope that you have understood all the topics which is discussed. So if you like this video, do like it and also share it with others. So I will meet you in my next class where we'll be learning about mitochondria and the plastid. So till then, take care and bye-bye.